Okay, guys, let's do it. Yesterday, the day before, we did prokaryotic gene regulation. Now we're going to focus on us, the eukaryotes, and how we regulate our genes. And just to put it plainly, we are complicated. And that doesn't mean that your work is going to be complicated. It just means that, in general, we are more sophisticated to figure out. So, if you could go and write on your heading, I'm going to write it up here in a second. Eukaryotic gene regulation. So on Wednesday and Thursday, we did prokaryotic gene regulation. We did operons. And I don't want you to think that this is going to be like a daunting task. Oh my gosh, he says so complicated. No, I'm just saying in general, we are more sophisticated. See, prokaryotes are simple. They're more efficient. They have less DNA, and therefore, their regulation is more efficient. If you have an easy job, I mean, it shouldn't be too much of a problem to do it well. But when it comes to eukaryotes, such as us, we have a lot of DNA, to say the least, and regulation is complex. Because there's just so much. It's like reading a children's book versus reading the Encyclopedia Britannica. It's a little complex. It's a lot more to take in. So I want to remind you all of what chromatin is. Chromatin is your DNA. It's just your DNA when it's all messy and disheveled and kind of like this big ball of yarn that's just a nightmare. I mean, this is chromatin. It's like a plate of spaghetti covered in spaghetti. That's chromatin. This whole thread is one strand. That is your DNA. Well, a chromosome is going to be that all densely packed. So chromatin is simply DNA that is decondensed. When it is condensed, it's a chromosome. Now, I have I want to try to really convey an idea to all of you, so try to follow me here and listen to the questions that I ask. I'm going to focus on 3 cells here to get this point across. First cell I'm going to focus on is an RBC, a red blood cell. If you're going into actual medicine, we call these erythrocytes. The next type is going to be a neuron. This is found in your central nervous system, your brain, and your spinal cord. And the last one I'm going to draw are smooth muscle fibers. If you didn't know this, we have 3 different types of muscle. And I don't mean like, oh, you mean like biceps, triceps, pecs? No, I mean like smooth, skeletal, and cardiac. Skeletal muscle is what you work out at the gym. It's what you run with. It's what you wake up and you're just so sore from your coach conditioning you and making you lift weights and running and doing squats and all that stuff. That's skeletal muscle. Smooth muscle is what lines your internal organs that are hollow, like your esophagus, your blood vessels, your stomach, your small intestine, your large intestine, and ladies, your uterus. That's all made of smooth muscle. You cannot control it. But it is a muscle, so it does contract. You just can't control it. And then the third type of muscle is cardiac muscle. That's only found in your heart. Well, the point I want to make to all of you is, if these are human cells, and they're also found in other mammals, how many chromosomes are there in the red blood cell? Jasmine? 0? Well, you're actually right. I was hoping no one would catch on to that, but you're actually technically correct. But let's just go with the fact that this is a somatic cell. Okay, so in general, how many chromosomes does a human somatic cell have? This is a number that every single one of you has to memorize. The number of chromosomes in a human. Jet? 46. How many chromosomes would you expect to find in a human neuron? Shane? 46. And Hartley, how many chromosomes would you expect to find in a human muscle fiber, which is a muscle cell? 46. Okay. Now, all of these are somatic cells. The definition of a somatic cell is a body cell. They have, in organisms, entire genetic code. So, Hannah, if I were to tell you that you have 100 trillion, let's see, trillions, billions, millions, thousands, hundreds. If I were to tell you that you have 100 trillion cells, how many copies of your DNA do you have? Yeah, you have 100 trillion copies of your DNA. Billions, millions, thousands, hundreds. You have 100 trillion copies of DNA in your body. Every single somatic cell in your body has all of your DNA. Okay? Now, Duncan, this neuron is in your brain, it's in your spinal cord. Does this neuron have the genetics to construct lung tissue inside of it? Yeah. Yes. Next question. Does this neuron, or let me rephrase it, is the gene to make lung tissue turned on? No. That's what I want you guys to understand. Every single one of your cells has all of your DNA, but that does not mean that all of your DNA is turned on. Risa, if you step on a thumbtack today, you're going to have a little tiny wound, which is probably going to bleed a little, so you need to make more skin cells, probably some blood vessel. Does that require you making bone? No. Do you have the genes to make bone present in your skin? Yes. Are those genes turned on? That is what I'm getting at. Good job. So let's just say that this is your entire 3 billion nucleotide long DNA. If you actually read one nucleotide a day, or excuse me, one nucleotide a second, it would take you 2 years to read the entire gene sequence. It's pretty long. Well, let's just say, for the heck of it, that this little section right there that I'm coloring magenta, that part will help you make red blood cells. This part that I'm coloring blue will help you make neurons. And the part that I'm coloring green will help you make smooth muscle fibers. Everything else, who cares? I could go on all day. I could say lung, I could say bone, I could say cartilage, blah, 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 blah. Now, Shane, I want to ask you, if I want to make red blood cells, which gene would be turned on? Do you remember which ones I said were which? The what? Yeah, this one, the purple one, is going to be on. Green light. Shane, what about every other part? Will it be turned on or off? That's right. Now, here's what I really want to convey to all of you. Shane, are those genes there? They are there. They're just not turned on. Right now, Andrew has a PS5 in his house. Is it turned on? But is it there? It's there, but it's not turned on, right? All right, so there's a difference between being present and being active. There is a difference. So that is what we're going to get to first. Chromatin that is densely packed chromatin that is densely packed cannot be transcribed by, and I think I've come a little short in telling you guys what this enzyme is, so maybe I'm wrong. You can help me out here, or you can prove my point. What enzyme turns, or doesn't turn, but what enzyme reads the DNA template to produce an RNA complement? Hannah? RNA polymerase. Chromatin that is densely packed cannot be transcribed by RNA polymerase. So the way that we phrase this, guys, is that the genes cannot be expressed. You can't get to them. If you have, and this is way beyond your time, it's beyond my time. People used to have a diary. And some of the diaries had a little key, and you'd have to unlock the diary. There was this little latch that would fold over it. You had to unlock it to open it up, and you could write, "Dear diary, blah, blah, blah." Well, you can't write in there, and you can't read the diary if the diary is locked. You can't feel, you can't read, you can't see what's covered up. So when your genes are very, very tightly packed, like this big wad of yarn, if it's so tightly packed like this, it cannot be transcribed. The DNA is just way too tightly packed. Can't get to it. So like if there is a gene buried way in here, RNA polymerase won't be able to get to it. So this term is known as, or DNA that is densely packed, or highly packed DNA is called heterochromatin. When DNA is highly packed, what made it so densely packed? It has been methylated. DNA that is methylated is practically inaccessible. You can't get to it. A methyl group is a CH3 group. So when one of those has been added to DNA, you really can't get to it. You just add that base to a part of the molecule that's readily available to have a bond. Yes, Duncan. Yeah, we're going to get to that. We are going to get to that. Now, you guys remember before Chris's break, we talked about something during sex-linked traits with Mendelian genetics. How many sex chromosomes do humans have? Oh, come on. You're overthinking it. We have 2. What are the sex chromosomes in males? X and Y. What are the sex chromosomes in females? Now, how many active X chromosomes does a male have in each of his cells? 1. How many active X chromosomes do females have in their cells? What do you call the X chromosome that has been shut down? Do you recall? Okay. A bar body. It has been methylated. You can't access it. And so males and females kind of have to be on an even playing field. So all you girls, you have 2 X chromosomes in every one of your cells, but one of them is shut down, and that is the bar body. So you have 1 active X, we have 1 active X. We're even. That has been methylated. It cannot be expressed. It cannot be transcribed. So if another concept that this relates to is something that we also discussed when we did Unit 5, and that's called epigenetics. Epigenetics simply says that just because your genotype says something doesn't mean that that's actually going to be your phenotype. The phenotype does not necessarily express the genotype. So if I were to look at, let's see. If I were to look at Addison, is that your natural hair color?
No.
Let's pretend it is. Next time say it. So Addison, is that your natural hair color?
Yeah.
Awesome. All right. Very helpful. So if I were to look at Addison's genetics, I would see that she is heterozygous. You all should know that. You see a dominant allele, you see a recessive allele. The dominant allele wins. Now, if the dominant trait was brown hair and the recessive trait was blonde hair, according to her genotype, what would you say her phenotype is just by looking at that? Brown or blonde? She would be brown. But when I look at her, she's blonde. What's up? Well, what if I told you that the dominant allele is being covered up? It has been methylated. It's still there. It just can't be expressed. What is the only other allele that is available? The blonde allele. That's the only one that's there. And so despite the fact that her genetics say brunette, 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 her DNA says blonde, 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 or because it's methylated. Her trait should be brunette. Now, can this be undone? Yeah. And so that's going to get us to my other point. DNA that is loosely packed is called Euchromatin. And no one has been able to tell me this today, despite the fact you all have taken this on your Latin tests. What does EU mean? You see it in eukaryote? Jasmine? It means true. This is true chromatin. DNA that is loosely packed can be expressed. So if you look up here at me, this is going to be euchromatin. You ready? That right there. You can actually get to that. It is now accessible. It's within reach. You can grab it. RNA polymerase can scan it, and it is going to be expressed. I see you raising your hand. Just wait. So rather than be methylated, this is acetylated. It's basically acetylated. It's basically been demethylated, which is the undoing of methylation. An acetyl group is going to be C2H3O. When you add one of those to DNA, it basically loosens it up. So RNA polymerase can express it. So imagine this. I have a chromosome. This black line I'm drawing, look how tightly packed I'm making it. This could be one of our 46 chromosomes. This is all made up of DNA. I mean, guys, this is pretty tightly packed. So I got heterochromatin. The other one I'm going to have is euchromatin. I'll highlight that in green. So all of this chromosome is basically heterochromatin. It is very tightly packed. Can't get to it. Except for, look at this. There's a little loop. It's a loose piece. This is going to be the part that's been acetylated, and you can actually access it. Now, Ayla, you had your hand raised. What's up?
So how is a piece like acetylated? Like, does it switch off or is it the same piece?
It has to, the acetyl group has to attach, and then you're either going to have a methyl group or an acetyl group.
So like, for like the neurons and the smooth muscle fibers, are those pieces like always different?
So let's ask, so, all right, that's a fair question. So let's make that a scenario. So let's go back up here. All right, Ayla, the purple part is the area that codes for red blood cells. The blue is neurons. The green is smooth muscle fibers. You have gotten into a car accident and you have a little bit of damaged neurons in your brain. You can fix it. It's not permanent brain damage, luckily. Which genes are going to actually be turned on? The parts that make red blood cells, the part that makes neurons, or the parts that make smooth muscle? So which one? So another way for me to ask you this is which one of those is going to be acetylated? All right, so we would say that that part is turned on. Now, do the other genes exist in your neurons? They do. Are they turned on? So we would call them methylated. Everything else is going to be basically shut off.
Is it always going to be, is there ever a time when the entire DNA sequence is turned on?
Not really, except during development when you're nothing but stem cells. Stem cells can become any type of cell. All of your cells were originally stem cells. And certain instructions tell them to become brain or bone or organ or skin or blood. Okay? Anything else?
So is that how you create like cells, like you, like from stem cells, like those cells?
So let me put it to you like this. There was a quarterback in the NFL for a while. His name was Peyton Manning. And he had to sit out an entire year of football because he badly hurt his neck. He needed stem cell therapy. He's an adult. The stem cells are really hard to find. And in your neck, you have your spinal cord. And that really doesn't heal very easily. That's why some people are permanently in wheelchairs and handicapped. So he went to Europe where they had treatments that were not available here in America. And they put stem cells in his spine. And it actually healed a lot of his injuries. The stem cells became spinal cord cells. So when you're a fetus, or not even a fetus, when you're an embryo, you are basically a bundle of blank cells. They don't have a designation yet. They can become any type of cell that you can instruct them to be. No, those are his stem cells. We have technology where you can find them and use them and use your own DNA, your own cells, and program it to become spinal cord or brain or whatever. Your bone marrow, they can be found in your umbilical cord. It was originally thought they can only be found in fetuses, which is why it was pretty controversial, because the only way to get a fetus or an embryo would be an abortion. So people were thinking, okay, there's the good and the bad. There's a whole ethics issue. But since that was like 20 years ago, but now we've been able to find them in adults. You can find it in your bone marrow. And so they used his own bone marrow stem cells and told them, they programmed his cells to become spinal cord cells.
And they would use the C2H3O to program them?
In a manner of thinking, yeah. It's a little more complex than that, which we're going to get to in a couple of weeks with biotechnology, when we get to that on the calendar. All right, now, let's get into how DNA is actually regulated. So when it comes to eukaryotes, there is a 5-stage process. So these are going to be the 5 stages of eukaryotic gene regulation. Eukaryotes, like us, really cannot multitask. We're not good at it. So we have to do one thing at a time. So the 5 stages are going to be as follows. At the same time, I was going to list them all out, but you'll just get the name of them as we go. Number 1 is going to be called the chromatin structure. Guys, in the chromatin structure, this is basically acetylated and methylated DNA. Remember that if it is acetylated, it is accessible and expressible. Methylated is inaccessible and inexpressible. You want to be able to get to the genes that can actually be methylated. So let's say, or did I say that wrong? You want to get to the genes that are acetylated. So I'm going to just make this blue line here. This is going to be your DNA, which is chromatin. Hope you understand that. DNA is chromatin. Chromatin is just when it's all squiggly and messy and not condensed nicely and tightly like a chromosome. And I just want to use this part right here. That's it. That is going to be acetylated, so typically green means go. Everything else is going to be shut off, and so everything else is going to be methylated, which means it will not be transcribed. So to say this one more time, I want to make sure I'm definitely getting my point across. All of your cells have all of your DNA, all of it. But that doesn't mean that it is turned on. All right? Now, that green region, green means go, that's going to be the part that will be transcribed. And by the way, I want to make sure I get this out there. If chromatin is DNA, and the first step is called chromatin structure, where is this happening in the cell? Where? The nucleus. If it involves DNA, it's in the nucleus. Okay, step 2. Step 2 is called transcriptional control. What is transcription? From what to what? You're using what to make what? You guys are frightening me. I know Jasmine gets it. I appreciate you wanting to participate, Jasmine, but I'd like to hear some other people to demonstrate that they know it too. Shane, you too. You two are 2 peas in a pod. Someone else. Lily, Casey, what is transcription? You use what to make what? You use DNA to make RNA. Why didn't you just say so? You use DNA to make RNA. That is correct. Now, I heard you say DNA, so where is this taking place? It is taking place in the nucleus. That's right. So transcriptional control is simply using RNA polymerase to transcribe a gene. Okay, so here's what I'm going to do, guys. If you could please look up. I'm going to zoom in on that right there. I'm going to copy that. Or not copy, but I'm going to make an RNA transcript of it. So here's that little highlighted green segment. There it is. That's the green part. Okay, we have 3', we have 5'. And once again, what enzyme transcribes this to make mRNA? RNA polymerase. So I'm going to draw RNA polymerase here. You probably cannot do this on your paper, so just watch. RNA polymerase is going to get on that DNA and go across it and produce an mRNA transcript. But the mRNA transcript is going to look a little weird. Here it goes. I prime end, 3 prime end. All right. The heck do I have blue and red there for? Because this mRNA is not ready. So we're going to call it primary mRNA. We have to refine it a little bit. Another name you could call this is pre-mRNA. Now, the red parts are called exons. For those of you that may have been doing your mastering, you may have seen these terms and you're like, well, how come we haven't learned these yet? Because we're doing them today. The red parts are exons. The blue parts are known as introns. From this point forward, I'm just going to put I and E to save a little time. So they're typically sequential. Exon, intron, exon, intron, exon, intron, exon. And that's it. That's it for transcriptional control. Well, what happens to those introns and exons? That's only step 2, guys. We have 5 steps. So let's go to step 3. Post-transcriptional control. Now, to save myself some time, I'm just going to drag this down here. You can redraw it. And I'm going to tell you what happens to those, a certain part of it. So we have to get this from pre-mRNA to final mRNA, Hartley. The first thing we have to do is we are going to remove the introns. The introns are basically junk. We don't need it. So I'm going to erase them. So we have an enzyme called a nuclease. Or not, yeah, nuclease? Yeah, nuclease.
What?
Nuclease is basically going to remove the introns. It's practically a pair of scissors. And so here we go. You ready? Gone, gone, gone. Yes?
That's like the DNA like it's using, and why would it get rid of?
Because there's a lot of parts of our DNA that is junk. When the scientists did the Human Genome Project in the 90s, they found that there's a lot of our DNA that codes for absolutely nothing. It's just junk DNA. Not all of your DNA codes for stuff. It's like reading the statement of agreement on an Apple iTunes form. There's just a lot of garbage that doesn't apply to you. So the introns are basically parts of the DNA, or in this case RNA, that really have no purpose. So you get rid of it. Okay, so part 2, the exons are spliced together. Now when you hear the word splice, you might think of slice. And when you slice something, you cut it up. That is not what it means. So don't let your brain go to bad territory. Splice means to bring together. There's actually a movie that's pretty weird called Splice. Anybody ever heard of it? It's the scientists create this monster freak show that's like part human, part fish, part plant, part bat, part everything, pig. And they spliced all of its genes together to make one organism. It's really weird. So the exons are spliced together. Splice means to bring together. And so what I'm going to do, if you guys just want to watch, check it out. There it is. We now have all the exons spliced together. We're still not done. Remember, we're currently trying to turn the pre-mRNA into final mRNA. I'm going to have to hustle because we have 5 minutes left. A 5' cap and poly A tail, which is on 3', are added. The 5' cap helps the RNA leave the nucleus. The poly A tail, which you can call a polyadenine, the A stands for adenine, helps the RNA survive in the cytoplasm. And so I'm just going to lasso my RNA. Come on. Bring it down here. And I'm going to add a 5' cap, this brown part, and I'm going to add a poly A tail over here. 5' cap, poly A tail. It is now considered a final mRNA transcript. It's all grown up and ready to leave the nucleus. And that's what it's going to do. It leaves the nucleus through a pore. The fourth step is going to be pretty quick. This is called translational control. Translation happens within a ribosome in the cytoplasm. So the location for this is going to be the cytoplasm. If I didn't write the location for step 3, that's post-transcriptional. That means it takes place in the nucleus. So this one's actually pretty easy. mRNA synthesizes a protein. I'm trying to get through this 4 classes over. So let's see, what color can I use? What color can I use? I'll use this one. This is a protein, but it is non-functional. I have never drawn a protein like that in this class ever before because it's not done. It's non-functional. It cannot do its job. And that's going to get us to our 5th and final step, I think just in time. This is called post-translational control. In post-translational control, the protein is going to fold and twist and bend into a 3-dimensional shape like this. This is how I've drawn proteins for you. So the protein is now functional when it is in a 3D form. This is known as the tertiary stage, which is a Unit 1 concept. And it is now capable of doing whatever it is this protein is supposed to do. Maybe it's to be hemoglobin or keratin in your hair or collagen in your skin, myosin or actin in your muscles. When it goes from being non-functional to functional, that is where it is finally complete. Now, there is a question I was going to ask you guys that I've seen in the AP exam. I didn't get to it in the 4th period. I'm going to try to get to it on Monday for you all because it's 2 questions that I've seen on the exam year after year, and it has to do with introns and exons.